Welcome. My topic is potato wart disease. Potato is one of the important tuber crop around the world, and wart disease is an important disease in the potato. It is first originated in South America. It is introduced from there to UK, and then now we come to see about the symptoms. First, above ground symptoms. Above ground symptoms are not usually apparent. We can observe like reduction in the vigor, some wart development on the upper stem, leaves, and flowers. The leaf also produces some minute wart-like structures, which is brown in color and then turns to black. Later on, it decays. Below ground symptoms: galls with varying shapes, mostly in spherical shape, around less than 1 centimeter to more than 8 centimeter in diameter. The ground galls are mostly white to brown in early stages, then later turn to black and they start to decay. These galls may also appear in the stem, swollen tips, and tuber eyes. At harvest time, they may be decayed. The tubers may be also replaced by the galls. They also develop after the harvest in the storage period also. It is not useful for table purposes. Now we come to see about the pathogens. Catum endobioticum is an obligate holocarpic endobiotic parasite. That is, it lives inside the tissues of the cells. It forms two types of sporangias. One is summer sporangia and winter sporangia. Mostly summer sporangia is spring sporangia. That is, it is active stage, and winter sporangia is a resting spores. It produces around 200 to 300 zoospores. These zoospores are posterior flagellate. Now we come to see of the disease cycles. During the spring season, the resting sporangia in the soil become activate and germinate in the soil and produce zoospores. These zoospores migrate in the soil, water, or while time of the irrigation or any time of the rain, and affects the epidermal cells of the growing plants. But the zoospores are short-lived, and so they insist inside the susceptible host for 1 to 2 hours. After the infection of the zoospores, the potato cells become enlarged and resulting in the characteristic wart galls. These galls are the source of the nutrient sink. These sporangias are produced sories, which are germinate to produce the new haploid structures in the susceptible host for secondary disease cycle. This cycle repeats and the gall formation becomes extensive invasion. Young galls are the nutritive sinks. First of all, the infection occurs at a susceptible host, which becomes a nutrient sink and rapidly expands to other plant tissues, such as the gall volume may be observed to be more and more, about 1000 folds in 16 days. We are going to see about the spread and survival. The spread in the soil by zoospores is limited. Mostly it will not spread, but the infection is be more. The zoospores lifespan is about 1 to 2 hours only. Long range contaminant like tuber movement in the international trade and soil with the plants and affected problem control soil. These are the main spread and survival for the zoospores. Favorable temperature is more than 20 degrees Celsius with high humidity. Winter sporangia may be dormant in the soil for about 20 to 30 years at a depth of 50 cm in the soil. Next, control measures. Mainly plant quarantine is one of the most important for the import sections. We can use also cover crops and wind-blown soils so that contaminated field soil cannot be transported to the normal healthy soils. Steam sterilization of soil is one of the major preventive measure. Soil treatment with formalin or mercury chloride is also advisable. Basically, the cultural method, which is the new tolerant or resistant varieties, can be used. For example, Kurfi Jyoti, Kurfi Kanchar. These are all the resistant varieties released for potato wart disease. I hope it is useful to you. Thank you.